Yeah, you got it.
I'm T-Jen, who's going to share a little bit about next generation sequencing, which I know is a subject a lot of us are very interested in learning more about. So, and you are all set. You
Thanks. Yep. Well, good afternoon. It's great to be able to have sort of a private chat with all of you in the room. Feel free to interrupt me. Is this feeding back a little bit? Feel free to interrupt me as I go along, okay? Because I don't care if I get to the end of my slide deck. I want to make sure you guys understand what I talked about. My task was to give you a bit of an update, a bit of an overview of what we can do in the research laboratory with genomic samples, DNA, RNA, and protein. And I sort of wanted to make sure we're all on the same page before I dove into that, because it's sort of a heady subject. So I want to start out at the 50,000 foot level with you guys to make sure you understand where we're at in the field and why we're here in the field right now, because it's actually a really important part of the story, okay? So probably about half of my time is a bit of an overview, and I apologize to those of you in the room who may know it better than I do. But I do want to make sure we're all sort of there, and I think it'll be helpful for the rest of the meeting too. Okay? So please interrupt if I lose you. That's not the goal. Okay, so it's my honor to speak to you today, but also to really tell some stories about our patients who we work with. I work at TGen. We have a clinic entirely devoted to the study of children with rare disease. I co-direct the scientific side of that clinic with my friend David Craig, and our medical director of the clinic is Vinod Narayanan, an excellent neurologist who we've worked with for a long time. Carrie Ramsey, sitting in the audience there, does all of our clinical research coordination, so the patients always end up loving her a lot more than any of us, which is the way it should be. It really does take a lot of people to work on a genome sequence and to do genomics work. And I'm proud to work alongside all these smiling faces every single day. When we discuss one of these cases, most of these faces are around the table giving their 2 cents about the data. And it's part of our secret to success is the fact that I can put the data in the hands of someone who's a pure wet laboratory person, someone who works at the bench with their hands, like Ashley. We can take this massive amount of data and put it into a form that she can digest. That's really, really important, okay? A lot of my colleagues in the field think that omics data should really only be analyzed by people who can do it using a supercomputer, okay? That's great. A lot of the upfront work is necessary to be done on a big computer. But being able to put it in the hands of as many eyes as possible has really been key to our diagnostic success rate. This is TGen, Phoenix, Arizona. We're a nonprofit. We've been around for about 14 years or so now. And really proud of our clinical space as well, which just opened in 2013, sort of shaped like a piece of pizza. Really creative painting and just a wonderful place for these families to come and visit and hopefully leave with a better understanding of their disorder and their disease. We have a big network of other clinics, other physicians, other researchers that we work with. This is also really important, as all of you know. This is how science and medicine really is done today and should be done. No one person, no one expertise exists at any one place in the world. It's really all about collaboration to get to these answers. Okay, so to date, our study, we've sequenced the genome of around 1,000 individuals. So that's about 300 families. And you can do the math and quickly realize that we just don't analyze the genome of the person with the disease. We analyze the entire family. This is truly an important part of the approach, and that's understanding the genetics of the entire family. We're extremely proud of our success rate, and I think that owes a lot to getting the data in the hands of as many people on our team as possible. So we are around about a 40% success rate for taking a family with no diagnosis and ending up with a gene that we think is the cause of their disease. So 4 in 10 who visit our center come out with that diagnosis. That's a big change. Much of it's due to what we can do now with the genome, actually being able to read that incredible book. Okay, so no questions on that stuff so far, right? Little Arizona nature images for us to recharge with. I'm a neuroscientist, and it's been shown that if you gaze at pictures of nature, you do get a recharge in your brain, so they're always in my talks. Okay, so we've been doing this a long time. I don't want you guys to think we just started in 2013 with the opening of our clinic. This is an old press release from 2005 where we announced our proud partnership with Dr. Narayanan to start doing this type of work. And back then, everyone in the field would admit we were throwing the wrong technology at all of these rare disease cases. We thought we had the right stuff, but it was nothing like what we can do now. And that's because these new machines that we use took us from this world of the analog device into a digital device. So around 2006, genetics finally started to do our work digitally. I think everybody can appreciate in the room how that shift is really, really important for keeping time. The same benefits are really, really important for genetics. So we don't use analog stopwatches to keep time at the Olympics now, right? We use digital watches that can measure it out to thousands of seconds. That's because of the accuracy, the transferability of the time, et cetera, et cetera. That's what we're benefiting from right now. That's why the next generation sequencers are playing such an important role in how we study disease. Of course, along with that comes an improvement in the cost. You guys have probably seen this slide before if you've ever been to a genetics talk before. It shows this massive drop in the cost of actually sequencing that human genome. So if it stayed up around here, we really wouldn't be having this conference today. Because it's dropped so much, we can do a lot more work, a lot more studies on the genome. And I sort of love this slide because it captures another reason why we're here today. So this is from 2012 at the inaugural Understand Your Genome event that was put on by Illumina, one of the leading companies that makes the sequencers that decode the human genome. And these are two different scientists. One on the left there, staring at an iPad, is a Nobel laureate. He received the Nobel in 1980, that's Walter Gilbert, for developing one of the ways to actually sequence DNA, okay? He was one of the chemists that developed exactly how we sequence DNA. We don't do it that way anymore, but he was really instrumental in jump-starting the field. On the right is one of the TGen scientists who looks at data all day. So he's an informatician. He just looks at zeros and ones and tries to interpret them to understand what's going on with the genome. This was a totally candid picture. Someone took it from across the room. But they're really interested in something here on the iPad, right? And you can probably guess what that is. That's actually Dr. Gilbert's genome sequence sitting on an iPad. Okay, so I just love this, right? Two different generations of scientists, both of them totally enthralled with really their first glimpse of a human genome reduced to something that was digestible on an iPad. So this is 2012. Of course, we do even more with this type of an approach today. Okay, so hopefully all of you already know this, but like I said, I just want to make sure we're all on the same page. So your organs in your body are made up of specialized cells. Inside each one of those cells is your DNA. And you can think of that any way you want. It's like your blueprint. It's like your instruction manual. You could consider it your personal book of life. But that DNA is, with a few exceptions, which we don't need to get into today, is the exact same from head to toe, okay? So that's why we can talk about in the previous session, do I need a blood sample or a spit sample to analyze your genome? Well, they're both should give me the same information, okay? All right, so how does a cell work? This is how a cell works in 30 seconds, okay? Sort of like a construction site, okay? So I imagine the DNA is sort of like the blueprint. So it's there in the foreman's trailer, protected from all the elements. In Phoenix, we do construction year round, and in the summer, if you don't have that blueprint protected in the foreman's air-conditioned trailer, it might melt out in the sun. That's sort of like the nucleus that protects your DNA in the cell, okay? And I imagine that every morning, the foreman meets with project managers to go over the blueprint for that building for that day and send them out with a plan with what to do for the day, okay? Those project managers are sort of like this sister chemical to DNA that's called RNA, okay? RNA is mobile, it can move, it moves out of the nucleus, okay? That's why the project managers come in, they get the plan, they take it back out to the rest of the construction site. Out in the construction site, the RNA is turned into protein, and protein does stuff, okay? That's all you need to know. Protein does stuff. Those are the workers out here actually laying the pipe and running the electrical wires and pouring the concrete, okay? So hopefully you can understand, just with this simple concept of how a cell works, if there's a problem with the blueprint, there's a misspelling or inappropriate mixture of concrete that's specified, the RNA, the project managers, take that information out to the workers, and the workers then make a bad building or lay bad concrete. Does that make sense? So that's how we can get disease, right? If we have a misspelling in our DNA, it ends up influencing RNA and ends up influencing protein, and that protein doesn't do the right stuff, okay? So hopefully you can see that that information's carried through. So in the research laboratory, we can do more than just look at DNA. We can look at RNA, we can look at protein, and it should tell us more information. Does that make sense? Okay. So as a geneticist, I love to ignore the environment, okay? It's like the hardest thing for us to study in science. But you all know it's really, really important, okay? So the environment is sort of like who supplies your concrete or cement. I always get those messed up. I think it's cement, right? So who supplies the materials that you then use to build your building? Those are sort of our choices. That's under our control. You know, do we exercise? Do we eat right? Do we drink to excess? You know, that environment is a little bit under our control. But what I want you guys to realize too is that since we can control that environment, there are some diseases that we can manage by controlling that environment, right? We know of a couple rare childhood diseases where if we control the environment, we can influence the outcome of something like PKU. Does that make sense? So even though there's a big genetic risk, if you control the environment, you can avoid disease too. Okay, great. Everybody with me? Okay, so how did we find places in our blueprint that were associated with disease in the past? Okay, we used a broken GPS. And let me describe it to you. So imagine if I told you I'm having a party. I can't take Saturday night, right? That's the gala.
Sorry.
So Sunday night, we're gonna have a party at my house in Phoenix, and I live here at this zip code, 85042. Okay, you're all invited. All right? That's really how we used to do genetics, okay? So there's a lot of information in 85042. You know that it's a planet Earth, it's the United States, it's Arizona, it's Phoenix, and there's the boundaries, okay? There's the boundaries of 85042. So I've got you pretty damn close to the party, right? But you still have a lot of work to do to find out exactly which house is mine. That's what we were stuck doing, just in 2005, 2004. This is what we did. So we would be able to find a region that was really, really important for disease, but we had such a difficult time finding the party, okay? So that's all gone now because we can read the entire instruction manual. We've gone from Cliff's Notes to being able to read War and Peace, as much as we all might like that in the room, okay? But that means that I actually just get the address and I go right there, okay? As long as I can interpret it, I go right there to the cause of disease. Does that make sense? All right. I don't want to make it sound too easy though, because it's not. So if I printed off your genome sequence, no tricks here, standard size, notebook paper, standard size, type font, and I stacked it up, it'd be about the size of the Statue of Liberty, 300 feet tall. That's 3 billion letters. And a lot of times we're looking for one single spelling mistake in that entire genome. So it's a lot of data. But one mistake can mean a lot, right? So here's my recipe for strawberry shortcake. And if instead of a cup of milk, I put in a cap of milk, just one single misspelling, I wouldn't make very good strawberry shortcake, right? So hopefully that makes sense. All right, so getting into the research part, I just want to remind you again about the construction site example and what DNA, RNA, and protein does. I'll give you some more different ways to think about it, okay? So organs made up of specialized cells, that's actually what a heart cell looks like, full of DNA, which is the blueprint. That's the way I think of it. Don't forget about its sister chemical, RNA. That's sort of what your cell decides to do, okay? Those are the project managers. You know, I ate a chocolate bar during the break and now I better digest that chocolate bar. That means I change these molecules of RNA to go do that. And then proteins are your cell doing work, the execution of those choices. All right. So the next generation sequencers let us look deeper than we ever have before. This is just a comparison showing that we can do that. It's really not important for you guys, but the point is we can look deeper and deeper in data than we ever have before because it's digital. But it's also much more reproducible. So this is 7 different experiments ran across 7 different months in my laboratory. So it's very repeatable. That's not the case with an analog device, okay? If I asked all of us, if we had a wall clock with this sweep hand and I said, okay, everybody, what time is it? We'd all have different answers, or some of us would. But if I just had a digital watch and I stopped it and I asked you to read the time, as long as you can read numbers, we'd all say the right answer. It's also helped us improve our speed. So we've gone from the first human genome taking 10 years to generate and analyze, and now we're doing it really in a few weeks. Under the most ideal, perfect situation, we can do it that fast. Okay. When did you want me to stop?
4:30.
How am I doing?
25 minutes.
Oh, plenty of time.
So you can bake us a cake.
Yeah, exactly. I wish. Okay, so let's go through a couple of research examples. So everyone now understands DNA, RNA, protein, at least at my sort of 3rd grade explanation level. You understand that we can measure DNA and RNA with the next generation sequencers. You've heard next generation sequencer thrown around before. It measures more than DNA, it also measures RNA, okay, which is really, really cool and really important. Proteins are measured with a different instrument, which we won't talk about today, but that instrument exists as well to measure proteins. So we can measure all 3 of those really, really important molecules. This is our center, c4rcd.org, if you guys want to learn more about us. We have an open study right now. It's open to try and diagnose families who don't have a current diagnosis. And Shelby Valent here, shown here, she was one of the first Arizonans to ever have her genome sequenced. She's our inspiration. We did her genome, I think it was 2010, and she just had an incredible result where her physician was able to change her therapeutic prescriptions. What he was giving her was changed based on the genomic data, and she's doing incredible today. It's really remarkable. I don't have time to do the story justice, but it's on there on the website if you want to learn more about Shelby. Okay, so we've heard discussion about a CLIA laboratory versus a research laboratory. So I'm going to frame that up for you guys here, and hopefully you understand a little bit better about what that means. So the discussion of a CLIA laboratory, there are rules around what can happen in that space. Doesn't mean that it can't change, but there's an approval process and a very rigorous testing process that has to happen to change really anything you do in that clinical laboratory space. Research laboratory, obviously, all bets are off, right? We can do almost anything we want. We can adapt to any new publication. So if a colleague publishes a paper on Monday, we can start doing that exact scientific approach on Tuesday. So there's pluses and minuses to both, which is why we've gone with a hybrid model. If I find something in the research laboratory, I can't go run and tell your doctor and tell you and really have it affect your medical treatment, okay? That has to be validated in a clinical laboratory. So that's great. We can do discovery over here, but it has to be validated over here. So here's our approach. We take in blood specimens, saliva specimens, whatever we're working with, into the clinical laboratory. So it stays in the clinical laboratory space when we receive the specimen. The clinical laboratory then purifies RNA and DNA for us, and then dispenses small amounts of it to the research laboratory. Okay, they sort of hand it across the threshold to the research laboratory. And then the rest of the sample is stored in the clinical laboratory. And then over here in the research lab, we do anything we possibly can to try and diagnose that disease. And if we find something that we think is possibly the cause, then we toss it back over the fence to the clinical laboratory and we say, will you test those samples you kept in the clinical space to see if you agree with us? And then they have to do it, you know, in their own way. But the beauty of this is the sample has stayed over here in what's called chain of custody in the clinical laboratory. So that means we don't have to go back to the family and ask for a clinical laboratory specific blood draw. So with just one single collection, we've been able to do research and then do clinical validation on it. Does that make sense? Okay. That's been our approach. Okay, now I'll give you a few examples. Are you with me so far? Okay. Make sure you interrupt or we're going to end up out of time. Okay, so what are some things we can do in the research laboratory that you may not have been hearing about in the clinical laboratory? First, we love to study families. A lot of times, if you're going to have a genome sequenced for a patient in a clinical laboratory, and if you're going to try and get it paid for by insurance, they're not going to sequence the whole family. They just want to sequence that one sick kid, okay? And that can lead to diagnosis, absolutely. But we have a much higher success rate when we look at the whole family. A lot of times we've got healthy, unaffected siblings, maybe some other sick siblings with the exact same disease, but also mom and dad. So this is an example of where you can see that by studying the family, we can see that the letter C here is the patient who had the disease. So this particular patient should have inherited this little green A from dad here, let's just say, in this hypothetical example. And the patient should have inherited this gray bar, which is a different nucleotide, a different base of DNA, from mom. And so it should be A and no A all the way down. But you can see with your eyes, this patient is only A. So what that means is they only inherited the copy from dad, okay? So we can use this as a significant clue. If we only had this patient's DNA, we may not be able to as clearly interpret that. In fact, there are lots of areas across the entire genome that would look just like that to our eyes if we only did the patient's genome. Does it make sense? We can also do something fancier that's called phased allelic expression. Okay, but you guys are all experts now in DNA and RNA, so this is an easy concept for you to understand. What this means is that we can tell if a patient is expressing at the RNA level, so if they're making RNA that's only from mom or only from dad, okay? So remember, we inherit one copy of the genome from mom and one copy from dad. When we go to make those RNA molecules inside our cells, it should be an equal mix of mom and dad. Does that make sense? But if we actually sequence the patient's RNA, we can look for shifts in that. So in some cases, you see that the patient might only be making RNA from mom at that one particular place in their blueprint. And that can help us understand disease, because what if mom was carrying a really bad change in her genome? She's healthy because she has the other part of her genome from her dad that she's using to compensate. But what if the sick child was only expressing mom's version, mom's damaged version, if you will, of that particular gene? You wouldn't know to really focus on that unless you were able to do this, unless you're able to determine which RNA molecule was happening from which parent. Does that make sense? All right, we're getting deeper and deeper. This is supposed to be a deep dive. All right, that's right. Okay, but this is really only, this is such a unique technique. This is really only possible today in the research laboratory, okay? You can't really put rules around this and execute it in the clinical laboratory space. Is that okay? That's why I want to make that distinction a little bit, okay? Not that it can't get there or won't get there, it's just early days. The other thing we can do is we can look at splicing. So remember, which we didn't talk about, so I don't know why I'm saying remember, but your genes are not made up as one big piece, okay? When they're formed into RNA and then made into protein to do the work in your cells, they have to get pieced together by some of your other cellular machinery, okay? It's like a jigsaw puzzle. To get it all to work, you have to put it together. There are some changes that can cause that process not to work very well, okay? And if that process doesn't work really well, you might leave out a piece of the puzzle that's really, really important for how that protein is supposed to work. And that's what I'm showing you here, and I'm calling it splice mutation detection. These are changes that can be seen in the DNA, in the blueprint, that alter exactly how these pieces are put together, okay? It could mean that if you're trying to build something that's really important for your cells, you might leave out a part of it. You might leave out 1/5 of it, okay? And you can really only see that if you're able to look at the RNA molecules in the cell. So the DNA molecules will give you a hint, but if you don't look at the RNA, you can't really convince yourself that it's happening exactly as you expected, okay? So this is another space where having your omics done in the research laboratory changes our ability to get towards a diagnosis. We can also look at copy number. So there are times when some of these diseases can be caused by deletion or amplification of a particular place in your blueprint, in your genome. So you can think of it like extra copies of my strawberry shortcake recipe in my recipe book, okay? Imagine if all of a sudden there were 10 pages of strawberry shortcake recipes right in a row, okay? Or, God forbid, imagine that strawberry shortcake was removed from my recipe book, okay? You can really detect that pretty clearly when you look at the DNA level. You can simply count how often you see particular pages. But that, again, is a little bit of an advanced analysis approach that's primarily only undertaken in the research laboratory. Okay? That can be really important for disease. Moving, I guess, above and beyond genomics, don't forget that we love to grow cells in the research laboratory too and see if we can help them, correct them. So one of the big things that we love to do is to study one of our actual patient cells in little petri dishes. We can understand exactly what their genetic cause is, and then we can try and rescue their cells, if you will, in a dish, okay? It's really a long-term goal here. This isn't something that is really easy to do. It's not easy to say, okay, these are sick cells, now let's rescue them. But it's one of the powers of being able to work with a research laboratory. Okay, so I think I'll just leave you with this quote, which is a pretty common one, but we love it for our center. And it's basically, hope is hearing the melody of the future, but faith is dancing to it. So this is realizing that the solution is already there. We just have to start to apply the tools that we already have. So we're really excited about the future, especially the future for rare diseases. And we are so honored to be a small part of the driving force behind it. Thanks. Yeah.
So, help me understand something, because I'm not very educated in this book.
Yeah.
My daughter has Alagille syndrome, and I work with the Alagille Syndrome Alliance. And it is known that in Alagille syndrome, it's an issue with possibly two chromosomes. It's identified which ones. So, is it something that we would advise our, let's say, our community, eligible community, to go get a sequence done to look for other things other than those two genes? Do we update this? Do we come back and say to researchers, is it really just these two? Is it more?
Great question. Great question. I think this is relevant for a lot of people in the room and at the meeting who missed this wonderful talk.
It started with one gene. Now we know it's two separate or together.
Perfect.
How does this?
Yeah, so that's a great question. So, and I think we're going to find this a lot. So back in the old days, we always thought that there would be a limited number of genes that would be the cause of any rare disease. I mean, if it's rare, come on, it's got to be simple, right? One gene. We're finding that's not the case. So on the research side, what we would love to do is to work with patients who have a disease that's supposed to have a problem with one or two genes, let's say, just to use your example. The first thing to do is the patient should be screened for those genes, right? So the first question is there a very clear genetic mutation, some change in those genes that is the cause of the disease? In most patients, we'll probably come back with an answer, yes, I have this change, right? But the ones who don't should then get genome sequenced, okay? So they're patients who have a phenotype that has led to the diagnosis of this disease, so that means they're phenotypically almost identical to that disease, but they don't have a mutation in any of the known genes. That means that there's probably a chance to discover something new or something outside of our current understanding for those two genes. Does that make sense?
It does. So a diagnosis for this disease could be done with, let's say, a liver biopsy to see how many ducts are working or not working. So it's not diagnosed with the gene. So do we say, okay, you've been diagnosed with the liver biopsy, you should go and get the genome test done, even though we would assume that those genes are involved?
Yeah, I mean, it's a great question. So I can guarantee you that the leading research labs in the world that care about this disease would love to screen anyone with a diagnosis across the two known genes. That's what they would love to do. I'll speak for them, right? Because they're looking for those patients that are negative across those two genes, because that's something new for the science. And that can mean a new therapeutic option. You're welcome.
I'm also new. Can you please explain to me how genome is different from exome sequencing? We're in the process of going through exome sequencing for big brain.
Great question. Okay, so the genome is the entire instruction manual, page 1 through page 3 billion, okay? The exome is a select number of pages from that entire large book. And the pages that are part of the exome are pages that scientists and physicians, people with the knowledge of the field, can most readily interpret, okay? So what I want you to realize is that we don't know much about our entire genome, but man, we actually do know a lot. So we don't know everything about those 3 billion pages, but we do know a lot about certain areas. And that's why people oftentimes start with the exome, because that's most interpretable, most easily interpretable. Does that make sense?
That involves DNA and RNA both?
No, the exome is still just DNA, but it's selected portions of the big book. And those portions that are selected out to be sequenced are the portions that we can most readily interpret.
And so if there is a problem with RNA or the protein, it's not
That's right, yeah, it won't. Yeah. And if there's problems outside of that particular exome sequence, it's not going to be detected either. But I just want to say that this approach makes sense, okay? Yeah. Okay, great. Yeah.
How far are we from being able to look at one specific gene that is showing all symptoms, that's been tested negative for DNA, but when you go back years later with RNA, is showing that there's mutations there and there's issues with that gene or the protein being expressed? So, like, for certain disorders where we know the gene that's most at cause,
Uh-huh.
how far are we from being able to help those families who are negative, but who show all of those symptoms, pointing researchers to say, we want to treat you like you are positive, but
Yeah.
you're negative right now based on research.
Yeah, great point. So just help me understand a little better about what you exactly want me to answer. So are you frustrated with the fact that if you don't get that DNA diagnosis, but you have the exact same symptoms, you still don't get the confirmed disease later? Yeah, and then you
We have families all around the world, but we have families in the U.S. who are not being treated as though they are positive, even though researchers who study that specific genes are positive that they are, they just can't prove it.
Right. Great question. That's a great question. I wish I had an answer. Because, I mean, I know why it's hard, because a lot of times patients are excluded from clinical trials simply because they don't have the DNA evidence that they have the disease that they know they have, right? I guess what I want you to realize is that we can, if you told me I care about disease X and it's caused by gene Y, and it's got to be something wrong with gene Y, can you study it at the RNA level? We can do that today pretty easily, in a pretty unbiased fashion, to see if there are problems with RNA. Yeah, but when I say we, I mean there's a lot of laboratories across the world that can do it.
Right?
Yeah.
So it is now more available than it used to be a few years ago?
For, if you said, tell me all about my patient's problem at gene Y at the RNA level, we can do that today using the sequencers, and it can give us a hint about if there are splicing problems or other things we may not have been able to predict 100% at the DNA level.
And then that would be interesting to compare those who test positive for it against those that test negative for it at an RNA level to see what...
Yeah, absolutely. So, when a disease is caused so strongly by a particular gene, oftentimes we see a lot of different flavors, I'll call them, of changes that lead to that disease. And some of those could be most clearly identified at the RNA level. Yeah.
So, my name is Grace. I'm with the National Alliance for Caregiving, and I have two questions that aren't going to be as sciencey. And the first is, it just strikes me like if I'm mom and I see that, I've been sequenced, my baby's been sequenced, my husband's been sequenced, and it turns out that maybe this is just me not understanding, but maybe it's my gene that's causing the problem. I think as a caregiver that there must be a significant amount of guilt and frustration around that. So I'm wondering if you can speak to, when you're talking with these families, how is that packaged? I mean, it's not just like a robot that rolls out, prints out your
Yeah, we
gene,
push the
like-
button and run away. And,
Yeah.
yeah.
So that's the first question, and then the second is, and that because we're across the lifespan, so we do a lot of work in the dementia space. So dementia, right? We don't know what the genetic factor. We don't know really anything about it except we know what we don't know right now.
Right.
So environmental factors, to what degree can this work tell us when the environmental factors switch on and off? And so like we know in dementia, if you exercise, if you eat the Mediterranean diet, there's some research that may stave it off. Again, thinking of a family, so if I know if I have kids, they may inherit some disease, there are environmental factors, we can tell them this type of work, how to switch that on or off.
Great question. Let's do the second one first. So, oftentimes the genetics can lead us to environment. So we can think about PKU as another example, right? That immediately tells us the change in this enzyme suggests we need to alter levels of this particular dietary thing. So I think that's maybe not a perfect answer to your question, but that's where I want you to think. We identify a lot of kids that have, man, this is feeding back. We identify a lot of kids that have mutations in biochemical pathways where it might suggest a dietary approach could help. So yeah, we are being informed about that, at least on the diet side. Your first question. A tough one, yeah. I mean, I think I would defer to my esteemed colleague.
Yeah, because I've worked with a medical director, and I think a lot of it, number one, is setting up expectations at the beginning of the research, which is involved in the consent process. So we have a section that talks about risks, and I always tell families that this is something that they may experience. And it's not, I mean, it's not going to change it. They're still going to experience it. They're still going to feel the guilt. But if they know up front that there's a probability that they may be carriers and they'll learn that information, then they know that where they come in and they know, oh, I might get that results back. And then I think it's also important, it is important, like when you're in the clinic, that you're meeting with the family, and I mean, you can do that over the phone, but that you're talking to somebody in the medical profession who's trained in getting back these results, or I mean, getting, like, genetic testing, and it may be a process. I mean, I spoke with a mom, and she was just, devastated about it, and so it took over a year, just talking to her, calling back, wanting more information. How does this affect the other kids that aren't affected? So it was just a process, and we never know how it's going to. I mean, I've had, moms who they say like, oh, I thought it was something that I had done. I mean, there's all these things that you never anticipate when you back adults. So in setting up expectations, and then having somebody that's trained and knowing that it could be more than just one contact with that person.
Yeah, it strikes me as, I don't know, I'll be real brief, so monopolize, but very unique to the rare disease community. And, in caregiving out of places, I can think of referrals that you would sort of send somebody to, but it's sort of scary to think about people having to deal with that without any resources.
But if I could just mention another anecdote, of course, sometimes it's also associated with relief, where the parents realize it wasn't something I did, during those 2 months after birth. It actually was this genetic change that was out of my control. So it cuts both ways. Yeah.
So I have a question and a comment, and I'll give you the question first.
Sure.
In so many of the rare diseases, we hear the term atypical. If it's an atypical presentation or a variety in the expression of the disorder, does either the RNA or the DNA look atypical?
Great question. It's going to vary for each disease, but so let's just talk generically about the way you could look atypical. It could be what we would call modifier genes, so other changes outside of the gene that is blamed for this disease that could be modifying your phenotype. Those are really difficult to find unless they're really, really rare. So we can have common modifiers, which we never would be able to really separate out. But does that make sense?
Yes.
Okay.
And then a comment. I feel like at these meetings, you're always preaching to the choir. Obviously, your team is fantastic, and you're doing all the right things. You have no idea how many of all of us have been on the other side of that coin. And I feel like there is, specifically with the rare diseases, it's like a foreign language. You're holding a baby in your arms, and a doctor
Yeah, let me see if I can talk myself through it.
Sure.
So this is a change that's encoded in her genome. So, and remember that genome's in every cell of the body. So there's little that, you know, so the goal in gene therapy would be to go in and replace that gene with a you know, perfect wild type gene. But that's really far off, really difficult to do. But I wonder if you've tried changing diet and sort of dialing back the chemical that acts at the receptor.
And what's interesting, what I did was I wrote down every change because she went from cytosine to thymidine and then a switch from arginine to cysteine. And in my research, I discovered with arginine, which she now has a lot less of, and it's proven in her amino acid test that we took that she's low on that, and cysteine is high. The issues that we see actually presenting in her physically are definitely a result. I can see what they say if you have a low arginine level, and also how vitamin A reacts with zinc. So now she has a zinc deficiency, and we found that out years and years ago before I got this diagnosis. We supplemented with zinc, and I'm so thankful because I realized we actually saved ourselves from a few major issues. So it does seem to make a difference in that, and that's kind of what I'm wondering if I can continue down that route. Is there a possibility, since she's technically still young, but I just didn't know if there's even a hope that could make a difference?
Yeah, I mean, you know, not knowing all the specifics, I think that what you're doing on the dietary side, remember, I'm just a scientist, you know, that makes sense to me. I mean, that makes total sense to me. And I think that the problem is she's young, she's going to go through all the other changes that we go through when we develop, so you might need to alter the diet a little more continuously until later in life. But that's what I would play it around with, because you know what acts, you know, what biomolecules interact with that receptor. So you can try to play around with those in the diet. That's what I'd be doing if I were in your spot.
Thank you.
All right, well, thank you, Dr. Henselman.